Thank you very much, and I'd like to thank also the organizers and for all of you to be here as well. So I'm going to try and talk about today about some of the strategies we're trying to develop in order to look systematically at the causal influencers of neural computations and behaviors. And a lot of our approach right now is to try to develop new technologies that we can use in order to try and dial in specific neural network states or specific activations of pathways or even simulations of certain receptors. And so what I'll talk about today is half will be about really the inventing of new technologies, including novel molecular reagents that we can use to try and activate and silence neural circuits, but also hardware that we're developing in order to try and deliver light into complex 3D patterns so we can start to sort of do the perturbation experimental analog of some of the recording, multi-electrode recording strategies you've heard today. The other thing I'll talk about is sort of the principles of how to control a circuit. When we control a circuit, even if we have great precision in the ability to activate or shut down circuit elements, there's also the network response, which can alter, sometimes in very unpredictable ways, what happens to the rest of the brain. And so we'll talk a little bit about how we're thinking about this problem and some of the new and unpublished studies that we're doing, both in awake mice and awake monkeys, to look at this. I should point out that on the technology side, we have a web page which has all the reagents and also increasingly white papers on how to design and develop the hardware. And also we've distributed the DNA and the viruses in collaboration with different nonprofits such as AdGene and the University of North Carolina. So the first part of this sort of three-part talk that I want to talk about is on reagents that we use to control neural circuits, activating them and silencing them. And control is important, of course, for many reasons. One is that it allows us to test a hypothesis. We can test the causality or the sufficiency or the necessity of a given cell type or pattern of activity in an output behavior or an output computation. Also, in principle, we could do therapeutic work as well. We could try to reprogram a corrupted neural circuit and to see if we can understand the principles of how to treat a disease by finding the most powerful points in a circuit on which we need to perturb in order to cause a beneficial outcome. Another important trend is that there's a lot of work that's emerging on defining new kinds of cells in the brain, different morphologies, different patterns of connectivity, different molecular markers. And if we could start to drive these inputs into defined targets within a network, that is also of importance. And then finally, temporal precision. If we can perturb things very quickly, that's important as well. So one of the things we've been working on is, can we simulate the actions of neurotransmitter receptors and ion channels where we have flow of charge across a cell membrane. That's what we're seeing in the schematic here, using light. And so here's a schematic of cells in a network. Each of these little dots is a cell in the prefrontal cortex loaded with a calcium dye. And if we can aim a beam of light at one cell or all the cells of a given kind, then we can start to play back activity to these networks. So I'll go quickly over the old and published stuff. The story begins really with a paper from Georg Nagel, Peter Hegemann, and Ernst Bomberg, where they showed that there existed light-gated ion channels. And in a collaboration with Georg and also with Carl Deisroth, I published the first paper on the use of these in neurons, where we showed that we could take a blue light-gated ion channel, Rhodopsin 2, which is usually found in green algae, put it into cells, and it would then cause the ability to activate these cells. So we could then cause this exact same neurotransmitter receptor kind of opening or ion channel opening with light, as I showed you in the previous slide. And importantly, it didn't require any chemicals for it to operate. It uses ubiquitous vitamin A derivatives. It lets in positively charged ions. It allows us to activate cells. And being a small piece of DNA, only codes for 300 amino acids, we could put this into a virus, deliver it into cells, and cause them to be expressing this molecule enduringly. And so to summarize a whole bunch of different papers, we first showed that we could activate cells expressing this molecule with light. So these blue dashes are the pulses of light, and the black traces are the spikes, and we could reliably elicit spike trains. We developed a variety of viral strategies, including lentiviral and adeno-associated virus methods, delivering these genes into cells. We developed optical methods for delivering light into the brain, using optical fibers attached to lasers. And since then, these tools have been used in a wide variety of different species. And today you heard about some of the other ones, but over the last half decade, people have used them in everything from worms to monkeys. And also transgenic mice have been developed that express these molecules enduringly in different cell types in the brain. And so this is an example of one of the first ones from Guoping Feng's group, which expresses these Rhodopsin 2 under the Thi1 promoter. And you can see, for example, that labels layer 5 pyramidal cells quite nicely. So what I'd like to talk about today are a number of new and directions we're heading in for developing neural silencers. And the story for this one begins quite some time ago from a paper by Okuno et al. In 1999. And what they showed in this paper is that if you plot the current versus the chloride concentration of various inward chloride pumps, then you could find some that would operate in the regimes that mammalian brains exist at. So these are Rhodopsins. They're light-driven inward chloride pumps from archaebacteria, and these archaebacteria use these light-driven chloride pumps to survive in very, very high chloride concentrations. And what you can see here is that a lot of these molecules, this is the Rhodopsin from H. salinarum, would pump chloride quite fine at high chloride concentrations, like 5 molar, but in the ordered rectangle, which describes the conditions in which the brain normally operates, you can see these molecules don't work very well. And that makes sense because these molecules are meant to operate in organisms that live in the Great Salt Lake, in Utah, and in different parts of the world, such as the Dead Sea, and so on and so forth. One of these molecules, however, was different from the rest. It had its peak operation at chloride concentrations that were comparable to those found in the mammalian brain. And this Rhodopsin from the Enophoronus archaebacterium we found was able to mediate hyperpolarizing currents, but they were modest in amplitude. Nevertheless, they did allow us to show until 2007 that we could hyperpolarize cells. And here you can see a neuron spiking away in vitro. We can hyperpolarize cells and shut down those spikes. And when we turn the light off, the spikes come back on. When we turn the light on, the spikes turn off. And so to first order, we can start to try and quiet down neural activity, although the modest size of the currents means that it's difficult to do this in vivo. So one of the things that we started to try and figure out was, could we mutate these molecules to make them better? And one of the reasons we wanted to mutate them was also that we found that there were kinetic issues with Rhodopsins. Here you can see a hyperpolarized neuron, but the hyperpolarization gets less efficacious over time. And then here we are giving during yellow light illumination, and now we're delivering little pulses of yellow light every half minute, trying to see if we can, if the performance will recover. But what you can see is the performance does not recover. Over a multi-minute period, the hyperpolarization still stays somewhat weak. We can deliver a brief pulse of blue light and cause the hyperpolarizations to return to their previous strength, but that complicates the optics somewhat because now we have to put in 2 colors of light for every point in the brain. So one of the things that a very talented freshman in the group, Alison Dobry, did was to screen through lots of different mutants of Rhodopsins, trying to see if we could improve the amplitude and if we can improve the time constant for recovery. If you actually look at the time constant for recovery of these hyperpolarizing influences to come back to normal, it really takes about tens of minutes to recover. And she was not able to find any one of these. You can see from the long column of no's in this column here. So another issue that's come up is that the Rhodopsins, at least in some cell types, seem to have alterations of cellular conductances at rest. So these are neurons from the hippocampus cut from slices and kept in the dark. And even though they're in the dark, the presence of the Rhodopsin alters the currents with respect to the resting state. And this is shown by a group last year. So earlier this year, we published a paper. The first authors, Brian Chow and Shira Han, that came out in January, where we were trying to screen through different genomic resources and to see if there were molecules that would have different hyperpolarizing influences that would be stronger. And one of the things that we found very quickly was that all of the Rhodopsins that we looked at had similar low amplitudes and similar poor kinetics of recovery, but the proton pumps that we found, these are outward proton pumps, which would decrease the potential of neurons by pumping positive charges out, actually had quite good performance. And so one of these molecules that we named ARCH, or Archaerhodopsin 3, was green light driven. It expressed very nicely in neurons, which is what you can see here. And it had about an order of magnitude better performance than the original Rhodopsin, both at low light powers, is what you can see here, and at high light powers. So what we did was we virally expressed these molecules in the awake behaving mouse. And what we found was that when we illuminated the cortex of these awake behaving animals, these spikes would decrease, the raster plot go quiet here, and then we turn the light off, the neurons reactivate. And here you can see dozens of trials where this occurs. And in fact, over half the cells were 99.5% or more silenced, which shows that we can get almost digital shutoff of the brain in the awake behaving animal. As you can see here from the population data, showing the number of cells that are undergoing different levels of quieting. The distribution is non-Gaussian, which makes us think that these might be the ARCH expressing cells, and these are other cells that are quieting as network activity declines. Now, we showed that this is a proton pump, And indeed, this is of importance because this is sort of an unconventional way of quieting neural activity. Protons are not endogenously thought of as the most common mediator of neural activity. And one of the things that we showed was that this was a safe method of quieting neurons. We started out by doing phenomenological experiments where we just shut down neurons for minutes at a time and showed that the activities after were comparable to the activities before. But we also did pH measurements using pH-sensitive dyes. And we showed that there indeed were small pH changes, but they were very tiny, 0.1 to 0.15 in magnitude, which is comparable to those mediated by the channel rhodopsins that we talked about earlier. This is measured by John Lin and Roger Chen in Biophysical Journal last year. And also it's similar to the magnitudes of normal variations in pH that people see in normal neural activity. So we also measured the passive properties of these cells and found that these cells were not changing their capacitance, resistance, or resting potential, which is important for concerns of cell safety. And finally, the recovery, which was very slow for halorhodopsins, where after we illuminated for a while, it would take a very long time for the performance to recover. We did not see such problems with these archaeorhodopsins. They recovered in tens of seconds, several orders of magnitude faster than the halorhodopsins did. One final advantage that these proton pumps offered was that they come in different color variants. So we found, for example, that there was a fungal opsin from Leptosphaeria maculans that was blue light driven, which if we compared to the original chloride pumps that were red light driven, we could actually quiet two different populations of neurons. One by, if we expressed the red light driven chloride pump, we could quiet it with red light, but blue light would not affect it very much. And then if we expressed the fungal opsin, which we called MAC, then blue light would quiet it down, but red light would not affect it very much. So this is of use if we want to try and shut down different neural circuits with different colors of light. Now, we've been screening lots of different genomic resources in the years since, and one of the things we've been looking for is more light-sensitive opsins. So this is a molecule that we've just submitted called ARCH-T, and we found this molecule is also very similar to ARCH in its performance in terms of peak current and so on, but it's about 3 to 4-fold more light-sensitive. And because it's more light sensitive, we think this could be quite useful to use in brains that are larger than the mouse brain. So this is a collaboration with Bob Desimone's group at MIT, where we're putting this molecule into the non-human primate brain. And what you can see here is that we have neurons in this case, parietal cortex, which are firing, and we turn green light on, these neurons shut down pretty much 100%, and then we turn the light off, they recover back to normal. And of the 45 single units we've recorded so far in the parietal cortex, we see a median 100% quieting in a 1 millimeter diameter ball centered at the optical fiber tip. We've also been working on trying to come up with other methods of silencing that might be more naturalistic. These methods, whether they pump protons or chloride or whatever, are pumps, and pumps are not the way that the brain usually quiets things down. The brain usually quiets things through channels, such as chloride channels and potassium channels. So we've been screening, and this is work done by Nathan Klepetke, Brian Chow, and Amy Chuang in the group. We've been screening for molecules that could more closely mimic potassium channels and GABA receptors. And so we've nicknamed this class of ion channels the Bizarro class. This is an example of one of the molecules, Bizarro 15. When we shine green light on it, we get a curve like this, where you can see it's a linear current voltage relation. This is current on the y-axis and voltage on the x-axis. We get a linear IV curve with a reversal potential around -50, which is not unlike that of GABA receptors, which are, of course, one of the major methods for neural silencing that happen naturally in the nervous system through interneuronal activity. Right now, we're starting to explore what happens when we shine different colors of light with different powers on the brain. Some preliminary data, although it's early, suggests that if we shine blue light on this molecule, we can actually get a linear IV curve with a shifted reversal potential that might be closer to that of potassium channels, although this is still earlier days. But it highlights our desire to find more naturalistic silencing molecules that could copy what happens in the brain normally and to go beyond what we can currently do with pumps. Now, I'd like to talk about some of the work that we're doing on hardware, because I think that hardware is very important if we really want to systematically start to address the three-dimensional densely wired complexity of the mammalian brain. And one of the methods that we've been working on is to try and figure out if we can deliver viruses into the brain in three-dimensional patterns so we can make entire neural circuits sensitive to light. So this is an example of some work done by Stephanie Chan when she was an undergraduate in the group. She developed fluidic injector arrays, which have multiple fluidic ports. Here you can see 3 of them, but we've started to design some that have even multiple dozens of injector ports. And when she injects this triple injector into the brain, she can label 3 independent points in the brain to make them light sensitive. And so our hope is that we can now try to perturb different parts of a neural circuit and to see how it influences other parts of a neural circuit. In order to do that, we've started to design custom 3-dimensional light sources. So the first attempt, we've been developing arrays of optical fibers, each optical fiber of which is docked to its own individual light-emitting diode. So here you can see a 14-fiber array that might be amenable to insertion into the mouse hippocampus if we want to tile a large portion of the mouse hippocampus. And each of these optical fibers that you can see glowing blue here is independently controllable, so that we can deliver light into the point that it targets in an independently controllable fashion. We've also shown that despite the large number of light sources that are mounted right here on the brain, we can put an electrode next to the optical fiber and do single unit recordings on neurons that are right there adjacent to these optical fibers that are on the light sources. These weigh about 1 gram or 2 and are about less than 1 centimeter wide. We're now working on ways to automate the insertion of these into the brain in order to understand if we can do 3-dimensional perturbation in awake-behaving animals. We've also developed wireless power and control so that we can power these in freely moving animals as they wander around. Also important because we are concerned and interested in making prosthetics to try and control the brain circuits that have gone awry, and wireless is important if we want to be able to do this in a chronic fashion and over long periods of time in a disease state. I also wanted to show some early data. In fact, I just made this slide a few minutes before the talk, where we're now trying to do true 3-dimensional control. So this is a drawing of a waveguide array where we have individual lasers at the top of waveguides, and these waveguides guide light down into the brain. And then if we go down deep into the brain, at the very end we have little mirrors that bounce the light out to the side. And the idea is that we could put this probe into the brain and illuminate points all along the probe in a way that allows us to address dozens of points with only the damage of a single optical fiber. And so here you can see some data that's hot off the presses here, where indeed here's a single probe about 300 microns wide that can emit light of different colors out at different depths along the probe. And so our hope is that we can start to develop arrays of these and to tile the brain in 3 dimensions so we can control each point in the brain with light and to figure out what each part is doing in terms of computation. Now, in terms of application, one of our hopes is that we can use these tools to really screen through circuits in the brain in order to figure out which ones are most important for a given therapeutic outcome or most important for a given phenomenon. If we need to turn off parts of the brain, we can figure out when they contribute to a given behavior. And so one of the areas that we're working on is to screen for targets that might be important for emotional processing. This is a classical example of fear conditioning, where a tone is followed by a shock, and animals learn that the tone is bad news. Over time, you can play the tone by itself, and the animals will grow at ease with the tone, but it takes some time. And so what we're doing is illuminating with light targets in the brain in order to see which ones we can use to accelerate recovery from this fearful state. And so here's an example where we're bilaterally putting in fiber arrays to target some of the classical known targets just to validate this procedure. The infralimbic cortex, for example, had been shown with electrical stimulation to be beneficial for alleviating fear states, but it hadn't been clear whether the cell bodies in this area were the most important or whether the white matter tracks that pass through are really the most important. And so what you can see here is that we can illuminate this area and just the cell bodies, and we can get the animals to relax faster in this tone extinction phase, whereas during the control animals, they do not experience this kind of alleviation of fear. One of the areas that we're really interested in is also to develop understandings of the principles of neural control. And I'd like to talk a little bit about some work we've been doing just in the last minute or so on non-human primates. One of the things that we're working on in collaboration with Anne Graybiel and Bob Desimone and other groups is to label excitatory neurons in the awake-behaving monkey so we can turn them on and off and to see what happens both in local and in distant cortical circuits. And our goal is to understand sort of the input-output functions of the cortex under manipulation, both because that's really important if we understand how the cortex works, but it's also really important if we understand the principles of how we need to be stimulating and controlling the brain to achieve a defined output in an algorithmic fashion. So what you see here is we developed viruses that in the monkey label excitatory neurons and not inhibitory neurons. We get about 100% labeling of excitatory cells and 0 overlap with GABA, the marker of inhibition. And one of the first things that we found, and we published this last year, was that we would activate excitatory cells, and many cells were activated, but also many cells were quieted. And so we were thinking, wow, this is interesting because it means that when we activate cells, we're seeing the driving of inhibition that then quiets other cells. And this is an important principle if we want to understand how to optimally control a neural circuit. This by itself might not be so surprising, but it was still nice to verify it. There were others who have shown using electrical stimulation that you get quieting of cells. Here you can see histograms where electrical stimulation causes quiet periods, but it's also nice to know that we can derive it purely from driving excitatory cells. Well, recently, we've been trying to figure out if this also happens with inhibition. So we've been activating cells, as I mentioned earlier, and seeing secondary effects of quieting. We've also been quieting cells down. This was the data I showed you earlier, where we showed that we can do quieting of neurons in the awake-behaving monkey. But we also see a small number of neurons that are excited during this process. So we're proposing now that we need to understand sort of this millisecond timescale homeostasis in the cortex if we want to understand the principles of how to control it optimally in order to achieve a defined outcome. And we'd love to start doing some computational modeling of this in order to understand how we need to drive the cortex in order to achieve a defined output state, which is important if we want to start to really program it like we would program a computer. Finally, I wanted to mention just one last closing bit. There's a lot of different disorders where neuromodulation, that is the use of neural control in the brain, is being explored. And at this point, probably close to 200,000 people, if not more, have some kind of neural stimulator implanted. About 100,000 people with cochlear implants, maybe another 50,000 people with deep brain stimulators implanted for Parkinson's disease, and many others who have spinal cord implants and peripheral implants. And one of the trends that's emerged, although it's still early days, is that certain virus types, such as the adeno-associated virus, seem to be relatively safe for delivering genes into the nervous system. The AAV, for example, the adeno-associated virus, has been used in hundreds of patients without a single serious adverse event due to the virus. It begs the question, however, about what these molecules, which come from algae and bacteria, do in the nervous system. And so this is my last slide. What we have been showing is that we can express these algal molecules and also these archaeal molecules that activate and silence respectively in the monkey nervous system. And we don't see any alterations in cell morphology. We don't see evidence of cell death or immune cell infiltration into the brain. And also we see no evidence of antibodies being produced in the body against these molecules as well. So although it's very early days, these are only a few animals, and it's still early days from a preclinical sense, we're very excited about the idea that perhaps these could open up molecular prosthetics that use light to control neural circuits in the future. So with that, I can take questions, I guess. Thank you.
Thank you very much for the amazing recent developments. So any questions with this presentation?
Yeah, I would briefly like to ask, you have spoken mostly about suppressing neural activity, but when you activate neural activity using channelopsin, how do you get around the problem of antidromic activation?
For antidromic activation? Well, there's a couple strategies, and it also depends highly, I think, upon the question at hand. One of the methods that we use, although it's somewhat primitive, is just to inject a certain region and then illuminate the cell bodies. There's some debate about whether axons are much easier to stimulate than cell bodies, and certainly in slices, people have found that axons are much easier to stimulate. There is a paper from Don Arnold's group at USC where they've targeted rhodopsin just to the dendrites in cell bodies using a myosin binding domain targeting sequence. And we've just started to try and play around with this and perhaps such strategies to reduce the expression or localize the expression might be the most powerful way to go in the years to come.
Yeah, so thank you. That was wonderful stuff. So I'm curious, you just mentioned about targeting to axons versus dendrites. How precise can you make your stimulation? Could you, for example, pick out individual axons in a bundle? Can you effectively stimulate axons in a fiber tract as opposed to cell bodies? How much precision do you have?
So a lot of work that on fiber tract stimulation has been done from other groups, not our group, but Carl Svoboda's group, for example, published some of the earliest studies on activating callosal axons across one hemisphere to the other. Axons are very small and hard to hit on a one-by-one basis. I don't think anybody's tried very, very hard to hit individual axons in an approvable fashion. Although more recently, people have been using pharmacological tricks to block, for example, as much of the surrounding areas as possible to suppress activity and then to photoactivate at a target to hope for suppression of other axons nearby. I think that it's unlikely that a pure optical method will be able to solve the whole thing, but a combination of optical methods with pharmacological suppression and also the ability to sculpt light. A group, Alapasha Vaziri at Julia Farm just published a very nice paper on sculpting light to try and hit neural structures very precisely. And that might allow the tuning of excitability of axons or dendrites by changing the light patterns in ways that would preferentially stimulate one or another. But that's still early days too as well.
And what about the two-photon approach? Do you have sufficient channel density now to be able to use two-photon illumination?
So the paper I just mentioned from Vaziri's group has been the most successful at getting two-photon stimulation by shaping the light pulse. So they're basically temporally sculpting the light pulse so that it hits the cell body, and that allows a maximal conjunctive activation, and they were able to get spiking in intact tissues. Another paper from Dave Tank's group has been able to do scan patterns that try and hit the membrane, and they were also able to get spiking, at least in cultured cells. So I think that a lot of people are now doing very clever methodologies at the optical level to sculpt the light in some way, either by moving the beam or by shaping the beam at a point. And the problem will probably be solved in the next year or two, I would imagine, completely. So.
Thank you.
Yes.
Great talk. I was particularly intrigued by your comments about activating excitatory cells has complicated effects, including suppression of other cells. Have you or anyone been able to selectively inactivate or activate inhibitory cells and see whether they have a different spectrum of effects?
Sure. Yeah, a number of groups have done that, but one of the questions is how much of the network you monitor. In other words, one of the big questions is here we are activating or perturbing excitatory neurons and recording those neurons nearby. It's hard to know for sure, though, whether other brain regions or other circuits are involved. Maybe these cortical neurons are sending, activation to a thalamic nucleus that has reciprocal effects on the local area. So although I think that there's a lot of interesting explorations of inhibitory neurons, and people are getting different results depending upon anesthesia state, whether they're doing in slice or in vivo and so on, what I think we need most is a way of really recording all the parts of these circuits so that we can start to look at the distributed activity.
But there's also such a great diversity of inhibitory neuron subtypes, and so this sounds like a great way to explore what are they actually doing that's different among that collection.
That's right. And also the positions of neurons as well are going to have a lot to say about what happens. Even a given molecularly described cell type, in layer 2, 3 versus layer 5 might have very different effects just because of their geometry. So.
Yeah. In the last part of the talk, you showed that in the cortical neurons, after the light was turned off, the inhibition still continues. But I'm trying several cortical neurons, and many cortical neurons shows rebound excitation, presumably caused by IH or low threshold calcium channel. Do you observe such kind of rebound excitation in your cases?
Yeah, it's a good question. So actually, I kind of went over the slide quickly, but you do see rebound excitations after the quiet periods.
Yeah.
When we activate excitatory neurons. For some reason, though, we don't see rebounds when we inhibit. And so one of the questions is, how does inhibition of cells let me be more eloquent here. How is inhibition not the opposite of excitation? Are there things that differ qualitatively in a non-bidirectional way when we perturb these circuits? And similarly here, we see rebound excitation after both the quieted cells and the excited cells, but here we don't see them in either. The time constants for recovery are also much slower. You can see it takes many seconds for the network to settle back down after an inhibition, whereas these are quieting back down in milliseconds back to their basal state.
But in our case, the rebound comes much earlier. So, but we are using much more general promoter. So I'm really curious whether some inhibitory neurons are also.
That's right. Yeah. Well, something we just started doing, although we don't have any data on this, is also to see what happens if we use a pan-neuronal promoter. Because if we think about using this tool sort of as an instantaneously reversible muscimol or baclofen, where we just want to delete a circuit just for a brief amount of time, deleting just the excitatory neurons might potentially, we don't know for sure, induce more side effects than deleting all the cells. And so one of the big questions that we'd like to figure out, or at least make some contribution to figuring out over the next year or so, is what happens if we just shut down all of the cells in a circuit as opposed to just the projection neurons, which might be very different from the way that neuroscience has been done in the past, using GABA, mucimol, baclofen, and other traditional methods of quieting circuits down.
Okay, thank you.
That's a great question.
Okay, thank you very much, Ed.
Okay, thank you.